All right, well, welcome to Lecture 2 for Biochemistry 501, in which we're going to continue talking about nucleic acids, and in this case, more specifically about DNA and how it's organized into chromosomes in your genome. And so we're going to be thinking about the stability of DNA and how that DNA molecule can be packaged and organized further so that it can fit inside of the cell, but still be used to access genetic information. And so, again, just as before, I'm going to post these learning objectives. If you can understand everything on these slides by the end of the lecture, you're in good shape. The key things we're going to be talking about today is one, the stability of DNA, why DNA is a better choice for storing genetic information than, say, RNA. And we'll talk a little bit about some ways of describing the stability of that structure. And then we'll also talk about how, even though you've got this great structure, in order to fit it in the genome, you have to kind of squish it down and package it in some way so that it fits inside the cell. And so we're going to talk about some of the topological properties of DNA that allow for this packaging to take place. And then we'll also talk about an important way in which DNA is packaged into the genome of eukaryotic cells, like you and me, by interaction with proteins to form a structure called chromatin, which ends up being the basis of making all of our chromosomes that define our genomes. And so let's get started talking about these issues. So as I said before, in the previous lectures, we learned that nucleic acids in general are really great for storing genetic information, copying, replicating that information, and transmitting it. So why choose DNA, though, and not RNA for this purpose? I mean, both of them have the Watson-Crick base pairing that is really kind of the essential feature needed to copy or replicate or access the information. So why go with DNA? And if there's a few reasons, but one of the reasons is that DNA turns out to be a much more stable polymer than RNA. And one of the reasons for this is that RNA, as you know, one of its defining features is the presence of a 2' hydroxyl group, whereas on DNA, there's just a hydrogen there. And it turns out that this 2' hydroxyl group can be activated for nucleophilic attack by bases. So we saw a similar we've seen this type of base activation of a hydroxyl to attack a phosphate many times before in the class. And in the case of RNA, some base can come along, like you see here, and activate this hydroxyl to promote attack on the phosphodiester backbone, leading to a break in the strand. So you end up getting this 2', 3' phosphoester moiety here, which can then be further hydrolyzed to completely break the strand. And so what this means is that an RNA molecule is more susceptible to degradation than a DNA molecule. And so obviously you don't want your master genetic storage to just sort of spontaneously break down. You want something that's more stable. And so definitely one of the reasons that DNA is used over RNA is because of this extra stability that comes from the lack of the 2' hydroxyl. Now, there's another reason that the DNA is a good choice relative to RNA, and that's something we talked about in the previous class, which is that the lack of the 2' hydroxyl in DNA enables a particular sugar pucker conformation, which you should know, which favors the formation of a B-form helical structure relative to an A-form structure that you see in RNA. And as we discussed, B-form DNA has one major advantage, which is that this sort of extended helical structure, like you see here, makes it possible for many aspects of the the sequence of nucleotides that are making up the helix to be read out through these large major grooves or minor grooves like you see here. And that's because, again, even though there are Watson-Crick base pairs holding A and T or C and G together, the major groove presents many possible hydrogen donor acceptor or hydrogen bond donor acceptor groups for interaction, and as well as the minor groove. And so this means that DNA, when it's completely sealed up, has the advantage that there can be many proteins that can, at least to some degree, read the contents of the helix without really needing to go open those strands up. Now, that's not to say that A-form RNA type structures can't have some information read through these smaller grooves. It's just that this B-form structure is really really optimal for this activity. So I mentioned, though, we're talking about how DNA is usually what is chosen. However, it is worth knowing that, on paper, there's no reason that an RNA molecule can't form the master genetic store. And there's a lot of hypotheses that in the early evolution of life, RNA was the original master molecule, and eventually DNA came along later. And certainly, in today's world, we often see that RNA is used as the genetic information store in viruses, which often have RNA genomes. And so many of you obviously have heard of the coronaviruses, which is why we're having our educational experience in this virtual way. That coronavirus has a single-stranded RNA genome, so not double-stranded DNA, single-stranded RNA. And there are other viruses like rotaviruses which have double-stranded RNA genomes. And so I just want you to keep in mind that although there's a lot of reasons why most sort of extant large organisms like us use DNA as our genetic material for the stability, the ease of accessing that B form, there's nothing that says that something like a virus or even some other living form couldn't possibly use RNA for this purpose. And that's just because all of those rules of complementarity base pairing provide a means of templating copying of one strand of information. So just keep that in mind. Now, one of the interesting things, though, about storing your genetic material in these nice B-form helices like DNA is that, those strands are going to exist in some, mix between perfectly double-stranded and annealed and strands coming apart into single-stranded structures. And so what's going to set the relative stability of the two strands that are being annealed together versus separated as free strands? This seems actually really important because the annealed form is how we want to store the material so it's safe, but we're also going to need to separate those strands from time to time to either be able to actually be able to make use of those base pairing interactions for replicating the DNA or transcribing certain stretches of the DNA into RNA. And so it's important for us to have a good handle on ways of talking about kind of the stability of these double-stranded annealed helices. And just to point out examples that you'll see in the class, every time DNA is replicated, you need to unwind the DNA into single strands, and so we need to be able to get in there to copy the material. And similarly, every time we transcribe a gene, what we're saying is that we're going to need to open that DNA double helix so that we can access one of the strands to copy it into a template. And so knowing about the stability of these, knowing about the stability of the double-stranded DNA helix is going to be helpful for thinking about how much energy it's going to take to open this up, or what types of things, activities the cell might need to have in order to accomplish this. And when you're thinking about the stability, this is one of those things where it's important to remember that not all of the base pairs in your double-stranded DNA are created equally. That is, you might recall from when we were learning to draw out the bases and the nucleotides and drawing these Watson-Crick-Franklin base pairs, we noted that AT base pairs form 2 hydrogen bonds, whereas CG base pairs form 3 hydrogen bonds. And obviously, you'll want to know which base pairs form stronger interactions so that you can keep this straight. And a useful mnemonic I use is that C and G are closer together in the alphabet than A and T, and therefore they form a stronger bond. But of course, you can memorize this AT2 CG3 however you like. But again, it's not just about memorizing some numbers, 2 versus 3. Each of those hydrogen bonds is adding some stability to the helix. And so because breaking or pulling 2 strands of DNA apart involves breaking those hydrogen bonds, we would expect that some DNA sequences are actually going to be easier to separate from one another than others, simply based on the number of hydrogen bonds that you observe. So if we had a sequence, for example, that was composed largely of A's and T's, so in this case, there are a total of 12 base pairing interactions here across A's and T's, giving us 24 hydrogen bonds. That means to pull these 2 strands apart is going to require us to break 24 hydrogen bonds. However, if instead we had a sequence made up of largely C's and G's, these 12 CG base pairs are going to give us 36 hydrogen bonds. And so to separate those 2 strands from another requires breaking 12 more hydrogen bonds, and it's going to be a lot harder to do so, and we're going to need more energy to do that. So, we can describe it in terms of the number of hydrogen bonds, but is there a more convenient way to describe the stability of these different double-stranded DNA sequences. And so a really simple method that's both very easy to do in the laboratory and then very practical with respect to a lot of laboratory techniques is the concept of the melting temperature, or which is called the TM for temperature melting. And that's the temperature at which, if you placed a DNA molecule in which the helix is half double-stranded, half single-stranded. So it's sort of the halfway point for denaturing this helix. And the melting temperature itself is related to the DNA stability. So if you have a very high TM, that means the molecule needs to be raised to a very high temperature in order to break it apart. And so you would think of those as really stable helices. Whereas if you have a very low TM, that means that it doesn't take very much thermal energy to cause the strands to fall apart, and those are going to be unstable helices. And so if we think back about those AT and GC base pairs, one of the things that they do is set the relative melting temperature of the double-stranded DNA that they're a part of. And so AT base pairs having only 2 hydrogen bonds compared to CG base pairs having 3 hydrogen bonds lead to a big shift in the apparent melting temperature of double-stranded DNA sequences that are otherwise the same length. So you can imagine that in this poly GC track here, we needed those 36 hydrogen bonds to disrupt this, and so its melting temperature is very high compared to a poly AT stretch over here, in which, because there's far fewer hydrogen bonding interactions in that strand, we have a much lower TM. And of course, real DNA in your genome isn't going to be all A's and T's or all G's and C's, and so different mixtures of A's, T's, C's, and G's will give you different melting temperatures for the associated genome. Now, in addition to it being a very useful way of measuring the stability of helices, it's actually very practical because many tasks that we perform in the laboratory to manipulate DNA are based on changing the temperature of different solutions of nucleic acids in order to get different pieces of DNA to anneal or interact with one another. And so the melting temperature tells us it's a practical guide for the temperatures that we need to go above to cause some fragment of double-stranded DNA to denature, and then the temperature we need to dip below to get 2 strands to anneal together. And so, for example, if we were in the laboratory and what we wanted to do was anneal this red sequence to this black sequence here, which was already annealed to another fragment here, if we calculated the melting temperature of this to be 72 degrees, then we would know that if we brought the temperature of this up to 72 degrees, these strands would start to fall apart. And then if we included this oligo here, we would need to bring, and we calculated that the TM of this interaction here was 38 degrees. We know we would need to bring the temperature down below here in order to get these 2 strands to come together. And so melting temperature is both really useful for describing the stability of DNA, but it's also something which is a very practical thing for us to use in the laboratory to think about the relative stability of different DNA structures. Okay, so to just sum up this part of the talk about the double-stranded DNA stability, our genome, so most biological genomes are made up of double-stranded DNA, and that's 2 single-stranded fragments of DNA that are annealed together through Watson-Crick-Franklin base pairing to form that B-form double helix. Many of the important tasks inside the cell, however, will require separating those 2 strands of double-stranded DNA into single strands in a process that's called melting. And the melting temperature is the temperature at which half of the double-stranded DNA is denatured. And this ends up being a useful, both useful descriptive as well as practical measurement of double-stranded DNA stability. And so if you get these concepts, 2 strands coming together, melting is the process of taking those 2 strands apart, and the stability of that interaction being related to the melting temperature, then you're in really good shape for understanding this portion of the DNA stability section of the talk. Now, in the next video, we're going to talk about other aspects of DNA structure and stability that have to do with being able to package that large polymer down. And to seed your thinking about this, you should note that if all the DNA in the genome were just one long, beautiful B-form helix, so no bending or anything like that, you would never be able to fit this molecule inside the cell. And so for some perspective, if you had an E. coli, the average size of an E. coli is about 20 microns. And the length of its extended genome, if you were to take all of its genetic material and stretch it out as one long helix, it'd be 2 millimeters, right? So that's a huge difference in terms of the size of the cell compared to the size of the genetic material that's packaging it. And things get even worse when you get into organisms like us, or in a blatant attempt to pander to you guys, this badger here, where the average cell size is 300 microns, and the length of its extended genome is actually 3 meters long. That is a huge difference between how big these cells are versus how much material you're trying to fit inside. And so obviously we're not going to be able to fit this material just as a long V-form helix. And so in the next video, we're going to be asking, how can the cell pack all of this DNA inside of itself to make it fit, and what constraints is that packing of the material going to have on our ability to melt the DNA strands and access the genetic information that's stored inside. And we'll learn the answers to those questions in the next videos.